Good morning students. In this video we are going to discuss about some important cell organelles in animals and plants. Those are mitochondria and plastids respectively. So in previous class we had a discussion regarding some information about cell organelles, and as we learn in previous classes, mitochondria and chloroplast, those are known as double membrane bound cell organelles in animal cells and plant cells respectively, right? So in this video we are going to discuss about a detailed explanation regarding mitochondria. First of all, let us see some information. Mitochondria, singular mitochondrion. Unless we use special stains, we are not able to see this mitochondria under the microscope. So we need to use some special chemicals, then only we could able to see this mitochondria even under microscope also. Okay, the number of mitochondria per cell is different depending upon physiological activity of the cells. Means you can see different cells in animals and plants, like muscle cells, bone cells, nerve cells, and in plants means mesophyll cells, parenchyma cells, and sponge parenchyma cells, etc. Right? So in different cells, different number of mitochondria are present. Those are depend upon physiological activities of the cells. Understand? Make sure this point must be remembered to everybody. So the number of mitochondria is vary from one cell to another cell. It's depend completely on the physiological activities of the cells. Okay, in terms of shape and size also, they are vary. It means mitochondria are vary even shape and size also in different cells. For example, a considerable degree of variability is observed in plant cells only. And we'll tell you one point here regarding the measurements of a typical mitochondria. Typically, it is sausage shaped or cylindrical, having a diameter of 0.1 to 1.0 μm, average 0.5 μm, and length 1.0 to 4.1 μm. Understood? Each mitochondrion is a double membrane bound cell organelle with the outer membrane and inner membrane. Understand? So the membrane which is present outside, it is called as outer membrane, and respectively we can able to see inner membrane also. You can see in this diagram on the screen outer membrane and inner membrane, and this outer membrane and inner membrane between distinctly visible space can be seen. The space is called as intermembrane space. That intermembrane space you can see on the screen. And one more point, outer membrane and inner membrane dividing its lumen, it means a space, distinctly into two aqueous compartments. So the whole mitochondria divided into two compartments. Those are what type of compartments? Aqueous compartments. It means some moisture nature can be seen. What are those compartments? Outer compartment and inner compartment respectively. Okay, now let us see about compartments in detail. The inner compartment is also called as matrix. You can see in this diagram mitochondria with two membranes. So the inner membrane and outer membrane respectively, and as usual entire space of mitochondria divided into two compartments, outer compartment and inner compartment. So the inner compartment is called as matrix. So the outer membrane forms the continuous limiting boundary of the organelle. So that is the role or nature of the outer membrane. It forms as a continuous limiting boundary of the organelle. Understand? But what about the inner membrane? The inner membrane also forms a number of infoldings called as cristae, singular crista. You can see number of infoldings towards inside on the diagram. So those infoldings are called as cristae, singular crista, and these cristae are present towards matrix. As we learn, matrix is nothing but inner compartment. Towards the inner compartment matrix, these infoldings are called cristae, are arranged in the mitochondria, within the mitochondrion. Understand? Now, some more information I would like to bring to your notice regarding cristae. Okay, what is the use of cristae? You can see in this diagram. Cristae increases the surface area of mitochondria. This is because the mitochondrion space is huge and the shape is also and the size is also vary from one to another, because the cristae helps in increasing the surface area. So when the surface area is increasing, then obviously mitochondrion appears somewhat big from one to another. When cristae is not having number of enfoldings, then the surface area will be decreases and it appears small. Understood? But the shape is same only, but size can be vary. So at the same time, I would like to bring to your notice one more important point. The two membranes have their own specific enzymes. Outer membrane and inner membrane, which you are seeing on the diagram. Those outer membrane and inner membrane are having specific enzymes, but they are associating with each other. At the same time, they are associating with even mitochondrial functions also. Generally, mitochondria exhibit some important functions. What are those functions? Can you believe that mitochondria is called as powerhouse of the cell? Because it is producing power, it means energy, in the form of ATP. In the form of ATP, full form adenosine triphosphate. So mitochondria is producing energy in the form of ATP by a process called as aerobic respiration, and it is also called as oxygenated respiration, and it is also popularized as cellular respiration. Then what is the definition for these terms? Now let us see one by one. Aerobic respiration. What is meant by aerobic respiration? The respiration which takes place in the presence of air, it means oxygen, is called as aerobic respiration. We breathe in oxygen, right? So that oxygen goes to mitochondria, especially into the cell the mitochondria is present. So into the matrix they goes and performs this process and producing energy in the form of ATP. So I repeat the definition once again, important for PU theory examinations. What is meant by aerobic respiration? Please make a note. The respiration which takes place in the presence of oxygen is called as aerobic respiration. So next, one more definition, cellular respiration. Please make a note this one also. Cellular respiration. The respiration which takes place at cell level is called as cellular respiration. I repeat once again, the respiration which takes place at cell level is called as cellular respiration. So this respiration required oxygen, oxygenated process, inhalation, understood. So they produce cellular energy in the form of ATP. Hence they are called as powerhouses of the cell. Understand? Now one more point. The matrix also possesses single circular DNA molecule. Understand? So apart from single circular DNA molecule, few RNA molecules also can be seen in the matrix. It means in the mitochondrion. So at the same time, ribosomes are 70S type of ribosomes. 70S type of ribosomes are present. Subunits 50S and 30S. Understand? And the components required for the synthesis of proteins. So as we discussed, ribosomes are also called as protein factories. Please make a note. Important for NEET exam and competitive exams case it also. And these ribosomes are synthesizing proteins since they are called as protein factories. Generally, the mitochondrion can also divide by a process called as fission. How come a cell organelle can produce new new organelles? How come it is possible? Generally cells can divide, but cell organelle is dividing here. That capacity is there to mitochondrion to divide new mitochondrion by a process called as fission. So with this context, mitochondrion is called as semi-autonomous cell organelle. Understood? So this is important information regarding mitochondria. So point by point, please make a note and write down, important for 5 marks theory examination also. Next, I am going to explain another important concept in this video. It is nothing but plastids. Plastids are generally found in only plant cells and euglenoids. We can't see these in animal cells. Understood? So plastids can be seen in plant cells and euglenoids. Example, euglena. These are easily observed under microscope as they are large in size. Understand? So if you keep one leaf piece under microscope, and you can able to see these plastids very clearly since they are large in size. They bear some specific pigments, thus imparting specific colors to the plants. If they are having green color causing pigment in them, then the leaf is appearing or the plant is appearing green color. Or if they are having yellow color causing pigment in the leaf, then the entire leaves and obviously whole the tree is appearing yellow color, right? If they're having carotenoid other pigments, then they appears pinkish or reddish color whole the leaves, and simultaneously the whole plant is appearing red color. So they are imparting the specific colors to the plants. Who are those? Plastids. Understand? Based on the type of pigments, plastids can be classified into three categories. They are, on the screen you can able to see, chromoplasts, chloroplasts and leucoplasts. Understand? So next, these again, these leucoplasts are again divided into three categories. What are those? Amyloplasts, elyloplasts and proteineoplasts. These proteineoplasts are also popularized as with another name, alleroplasts. Understand? First of all, I would like to explain about chloroplasts. The chloroplasts contain chlorophyll and carotenoid pigments, which are responsible for trapping light energy essential for photosynthesis. Understand? So you can able to see chloroplast on the screen. This chloroplast is imparting consisting which color? Chlorophyll and carotenoid pigments, which are responsible for photosynthesis process. By trapping the sunlight, light energy is converting into chemical energy. So during this process, synthesizing energy starch. Understand? So this is the function or duty of these chloroplasts. Then in, let us talk about the second one. In chromoplasts, fat soluble carotenoid pigments like carotene, xanthophylls and others are present. Understand? You can see chromoplasts on the screen, and this gives the part of the plant to yellow, orange or red color. Understand? So yellow, orange, red color is due to because of chromoplasts. Next, third one, you can see on the screen, the leucoplasts are the colorless plastids of varied shapes and sizes with stored nutrients. So shapes and sizes are different, but the main role of these leucoplasts nothing but storing the nutrients. And one more point, they're not having any color causing pigment since they're called as colorless plastids. Next, let us go through very detailed information of leucoplasts. Leucoplasts are having three categories again: amyloplasts and elaioplasts and aleuroplasts. Okay, now let us see role of each one. Amyloplasts stores carbohydrates. You can see on the screen. So when they are storing the carbohydrates, nothing but starch, they can easily identified in many plants like potato, sweet potato, etc. Then second category, elaioplasts. They stores oils and fats, so they can seen in many plants like groundnut, sunflower plants, etc. Next, aleuroplasts or proteinoplasts. Their role is to store proteins. So these plastids can be seen very easily in plants like beans and grams. It means pulses. Understand? So these are about leucoplasts. Now students, in this video only, we are going to discuss about in detail regarding chloroplast. This chloroplast structure and explanation is also very important for 5 marks, like mitochondria. So from this chapter, the cell unit of life, these two cell organelles, mitochondria and chloroplasts, both are very important for theory examinations, each one for 5 marks category with a detailed labeled diagram. Understood? So now I am going to explain chloroplast. Majority of the chloroplasts of the green plants are found in the mesophyll cells of the leaves. Understand? So these are what kind of shape are having. These chloroplasts are not having same shape in all the leaves. They are vary from one to another. Even lens shaped, oval shaped, spherical shaped, even discoid or ribbon like structures also sometimes having variable length. Even generally the way how shapes are different, even length is also different. Now let us see length. 5 to 10 mm, that's why we can able to see very clearly under microscope, and width is 2 to 4 mm. Understand? So their number varies even from one to another cell. For example, I would like to bring two important examples to your kind notice, which are very important for NEET examination. Let us take Chlamydomonas. Chlamydomonas is unicellular organism. Since it is unicellular or single cellular organism, only one chloroplast is present. So this Chlamydomonas is example for one per cell chloroplast of the organism. Understood? Next, green alga. Generally, this green alga consisting 20 to 40 chloroplasts per cell in the mesophyll cells. So each mesophyll cell has 20 to 40 chloroplasts. As we discussed, in mesophyll cells, chloroplasts are present majorly. Next, one more point regarding chloroplasts. Like mitochondria, the chloroplasts are also double membrane bound cell organelles. You can see on the screen outer membrane and inner membrane. Outer membrane is a boundary and inner membrane is the lower one and presented below the outer one. So here one of the two, the inner chloroplast membrane is relatively less permeable. Understand? Between these two membranes, inner membrane is relatively less permeable and the space is present between the two membranes. At the same time, the space is also present inside the inner membrane of the chloroplast is called as stroma. So you can see the stroma on the screen. The space which is present in the chloroplast below the inner chloroplast membrane is called as stroma. Please make a note, this is very important. Next, one more point regarding chloroplast structure. We are discussing structure of the chloroplast. Listen to me very carefully. A number of organized, flattened membranous sacs are present. They are called as thylakoids. You can see thylakoids on the screen, right? So what kind of structures those are? Those are organized as a flat structure and membranous sacs. Membranous sacs means inside some free space is present. That free space is covered with a membrane, hence called as membranous sac-like structures. That too, they are flat in nature. They are called as thylakoids. You can see thylakoids membrane on the screen. Then one more point. Thylakoids are present in the stroma. Understand? But thylakoids are arranged in stalks like the piles of coins called as grana. You can see on the screen thylakoids one above another arranged, and the set is called as grana. You can see grana here. Singular granum. Singular granum. How they are arranged? I repeat once again. Thylakoids are arranged in stalks like piles of coins called grana. Singular granum. Or the intergranal thylakoids. Grana is also called as the intergranal thylakoids. Understand? Next, one more point regarding this structure of chloroplast. In addition, there are flat membranous tubules called the stroma lamella. You can see stroma lamella in this diagram very clearly. So one granum is joining with another granum with the help of membranous tubular structures. Those are called as stroma lamella. So you can see stroma lamella here on the screen in this diagram. Next, and this stroma lamella role is connecting the thylakoids of different grana. Connecting the thylakoids of different grana. By whom? Stroma lamella. So next, one more point. The membrane of the thylakoids enclosed a space called as lumen. You can see lumen. The lumen, which is also called as space, the membrane of the thylakoids enclosed a space called as lumen. Understand? So you can see very clearly in this diagram. And the stroma of the chloroplast contains enzymes. So you can see stroma in this diagram contains enzymes required for the synthesis of carbohydrates and proteins. You know, during photosynthesis, this chloroplast, which is popularized as kitchen of the cell, is performing photosynthesis process, right? And synthesizing carbohydrates, it means starch and proteins. So to synthesize the carbohydrates and proteins, there are few enzymes are present in the stroma. Understand? Next, one more point. It also contains small double-stranded circular DNA molecule and ribosomes also. Where are those present? In the stroma. You can see on the screen double-stranded DNA molecule, that to which shape? Circular. See, circular shaped structures are there in this diagram, right? What are those? Those are double-stranded circular DNA molecules, and a few RNA molecules, few ribosomes are also present. So these ribosomes are having specific characteristics. Now you can see one more important point in this diagram. Chlorophyll pigments are present in the thylakoids as we discussed. So where are the chlorophyll pigments are present? On the thylakoid membrane. So you can see thylakoid here. They are dark green in color. Why they are green in color? Due to presence of chlorophyll pigments. I repeat once again, chlorophyll pigments are present in the thylakoids. I repeat one more point here. Double-stranded circular DNA molecules and ribosomes are present in the stroma, and as usual, chlorophyll is present on the thylakoid membrane. Understand? Next, ribosomes. What type of ribosomes are present in this stroma of chloroplast? Ribosomes are generally small in size. There are 70S type of ribosomes. So subunits are 50S and 30S, and they are smaller than the cytoplasmic ribosomes. See, this chloroplast is present in the cytoplasm of the mesophyll cell, right? So in the cytoplasm also ribosomes are present. What are those? 80S type of ribosomes. Subunits 50S and 40S. As we discussed, ribosomes are present in different places. What are those? In the cytoplasm. Next, in the mitochondria, in the chloroplast, and as usual, on the rough endoplasmic reticulum, and their duty is protein synthesis. Understand? Since the ribosomes are present in the stroma, proteins can be synthesized. Then, since the enzymes are present in the stroma, carbohydrates can be synthesized with the help of chlorophyll, which is present in the thylakoids. Understand? So students, hope you understood this mitochondria and chloroplast. Thank you one and all.